It's an honor to be invited to speak to this group. I want to tackle two tasks. Today, I want to speak briefly about the ITP and bring you all up to date on that. But then I want to spend the last 15 minutes or so of the talk on a new idea, the idea of aging rate indicators, a kind of speedometer for how rapidly you are aging, which I think has the potential to be of great value to aging science, including translation to the clinic. This chart shows why, if you're spending your life looking at cancer or diabetes or Alzheimer's, you should instead be working on aging research, because it is much more likely to be efficacious in terms of producing long, healthy, active lifespan. An average woman lives to be about 80 nowadays, and if you cured cancer completely and no one ever got cancer again, her lifespan would go up only by about 2.6 years. It's true also for heart disease and cancer together, but rapamycin by itself in mice can do about 6 times or 9 times better than that in terms of percentage increase. So anti-aging medicines are where the action will be in terms of practicalities for human preventive medicine. There are 4 new concepts that I think have been established in the last couple of decades. The first is very important, that aging can be slowed in mammals by diets, by single gene mutants, and by drugs. I have an hour talk on this, which I won't give to you because of the pressures of time today. But it's very important to know that aging can be slowed. If you, as most people, believe that it can't be slowed, then aging biologists are wasting their time. And doing so delays many lethal and non-lethal diseases. Some people will say, oh, I don't want lifespan, I want health span, as though you got a trade-off. You could have one or the other. But that's backward thinking. In fact, things that promote lifespan in mammals do so because they slow the loss of health. That is, they also extend health span. The two are linked together and not mutually exclusive. The studies, which I will briefly touch on, have given lots of new molecular clues, paying attention to IGF-1, to uncoupling proteins in the mitochondria, to mTOR, of course, changes in glucose. These give us good leverage for beginning to look for new anti-aging drugs. The old paradigm, the thing that was how researchers thought about this for many years, and too many people still do, is to think that it's important to ask what causes aging. It is not important to ask what causes aging. It is important to ask what slows the rate of aging. The thing that causes aging in the eye and in the brain and the bone and in cancer cells, those are very different, very heterogeneous. And controlling them all one at a time is kind of a fool's game. You can't really get much done. Figuring out what controls the rate of aging is where the action is, and that'll be the point of the second half of the talk. So let me give you an introduction to the ITP. This is insanely abbreviated. I mean, this really deserves an hour or so, but I wanted to touch upon it very quickly so that I would have time to talk about the aging rate indicators. The key features of the protocol are that we would never, ever, ever use inbred mice. We would never use black 6 mice or any kind of experiments where all the mice were the same genotype. So we use genetically heterogeneous mice. Any 2 mice in our study are brothers and sisters. They share half of their genes, just like you share half of your genes with your brother or sister. Anybody, which includes you and you and you and you, can suggest a drug. There's an annual please send us your ideas competition. There's a set of review committees that look at these, and then we pick 5 or 6 or 7 a year, and you become then a collaborator with the ITP. At this point, several hundred scientists have suggested drugs to us. They're not tested just at my lab, they're tested at 3 labs. Mine at Michigan, Ron Corstange's at the Jackson labs, and then Randy Strong at Texas, so that if something works at all 3 labs, we have more confidence that it will really work in your lab. And we use an awful lot of mice. For each drug that is selected, we have 300 drug-treated mice and 600 control mice. And the reason for doing that is we want to be able to detect 8% or 10% changes. Now, 85%, this is not surprising, 85% of the drugs that were suggested to us by very smart, very educated, very sophisticated scientists, they do not work. It's very hard to guess whether a drug is going to extend lifespan or not. I've put into red the famous ones, the ones that have been mentioned in the popular press: curcumin, resveratrol, fish oil, nicotinamide, riboside, the alleged senolytic fisetin, metformin, a very famous and terrific diabetic drug. None of these extended mouse lifespan. But at this point, 12 drugs have worked. The most famous of these known to you is rapamycin. This is our follow-up experiment, and you can see that in both male and female mice, it leads to dose-dependent increase in median and maximal survival. This is the summary slide corresponding to those survival curves I just showed you. At the highest of the tested doses, male lifespan was increased by 23%, and female lifespan by 26%. So to again put that into context, if you cure cancer in people, you get a 3% lifespan increase. So this is a very large effect. It was an extremely pleasant surprise to find that rapamycin works just fine if you started in middle-aged mice. I am myself, well, I was middle-aged at the time this experiment was published, and many of the people who pay for this work are middle-aged, and many of the people who run the government are middle-aged, so they're always interested in drugs that work in middle age. I predicted incorrectly that the drug would only work if you started it in youth. But that's wrong, and this is very good news for those of us who are no longer fully youthful, but would like to find some way of slowing the aging process down in a meaningful way. In the mouse, at least, of course we don't know for people, but in the mouse at least, starting in young adult mice and starting in mice just at the threshold of old age has exactly equal effects. You can't tell them apart. And in fact, of the 4 drugs that have been tested in this way, 3 of them show that characteristic. Doesn't matter when you start. And the other one works half as well. Acarbose works half as well if you started in old mice as if you started it in young mice. That, to me, is the biggest experimental surprise, and of course it's very good news in terms of those of us who actually want to have an impact on human health. Now, these mice, though they mostly die of cancer, it's not just that rapamycin is a terrific anti-cancer drug, but in fact it slows the aging process per se. This is some of that evidence. Liver changes with age, heart changes with age, uterine changes with age, adrenal changes with age, tendon changes with age. We had in this original paper 10 separate kinds of age-dependent tissue changes. 9 of those 10 were slowed down by rapamycin. So although the thing that actually accounts for the lifespan shift is its astonishing ability to delay all sorts of cancer, blood cancer, breast cancer, lung cancer, liver cancer, in fact, it is doing that by slowing the aging process. This is the summary slide. I'll leave it up for just a moment. And of course, if you want a copy of any of these slides, just send me an email and I'll be glad to send them to you. This summarizes all of the drugs with the published ones at the top, the unpublished ones at the bottom, that so far have had a significant effect either on males or on females or on both on lifespan. Everyone here has probably heard about the rapamycin work. The other 3 ones that we published early were acarbose, canagliflozin, which is a member of a class of anti-diabetic drugs, and a weird steroid called 17-alpha-estradiol. A little bit further down the list, you can see meclizine and astaxanthin. They're worth mentioning because they are available, at least in the United States, without a prescription. They are the first over-the-counter medicines that you can purchase today, if you want to, which have met this test in mice. Now, of course, we don't know if they would be workable in people, and we don't know if they would be good for people. We have no way of knowing that until someone actually tests them and finds out. But they work in mice. And then the bottom 4 lines are ones that were either just published in the last year or the 2 new ones that will be in our paper that's coming out next year. MSDC 160 is an insulin sensitizer, and halafuginone is a mitochondrially active drug. So there are lots of different things that these drugs do. Their primary targets are quite different. And that prompts the question and gives the tools to ask what I view as the really most important question: are there shared mechanisms? Are there shared mechanisms that could potentially point us to the target that really counts? So this summarizes the ITP part of the talk. 6 bullet points. People who think aging can be slowed are wrong. There are a lot of them. Many of them are keen scientists, but they got that wrong. and they ought to start changing their minds. Many of these drugs, for unknown reasons, are sex-specific. Of course, we don't know if they would work in people, and if they did work in people, we don't know if they would be sex-specific. But in mice, somewhat to my surprise, some work in males, some in females, and only a minority work in both sexes. I've mentioned they work in middle-aged mice, which is really good. I mentioned they work by extending health span. You don't just get a crippled, sick, demented, cancer-ridden mouse which won't die. You get a mouse that postpones all the bad stuff, and they give specific leads for new molecular indices. But these provide key tools for aging rate indicators, which is the rest of the talk, and give us clues as to shared anti-aging mechanisms. So the problem is, as I'll bet you've guessed, it is not easy to guess what drugs will extend lifespan. The best scientists in the world guess that wrong 85% of the time. So the challenge we set ourselves was to find fairly quick ways that is quick with respect to a mouse lifespan, that would improve the frequency of good guesses. And so we had this approach. We had, at the beginning of the study, 9, now 11 different kinds of mice that were slow aging, but slow aging for different reasons. And we asked, what is it that these all have in common as young adults? Are there any shared pathways, common pathways, that you will have if you're going to age slowly, whether it's a gene, a diet, or one of these drugs? So here's the basic strategy. We had 4 kinds of slow-aging mice: Ames, Snell dwarf, Pape, knockout, and growth hormone. We've added P10 overexpressors. That's published now. 1 anti-aging diet, the famous calorie-restricted diet. We've added another with Dudley Laming's collaboration with isoleucine-restricted, and 4 drugs so far: rapamycin, acarbose, 17-alpha-estradiol, and canagliflozin. So the goal was to find changes induced in young adults by all of these mice. So let me give you an example, and this one I'll walk you through somewhat slowly because the same format will occur for the next 4 or 5 slides. We're looking here at a protein. It's called GPLD1. It's in the liver, it's in the plasma, and it's in the fat of mice. Others, Saul Villeda in particular, had shown that if you exercise, you get a lot of this. And in fact, if you exercise and you get a lot of this, you get better cognition. So it is probably one of the key mechanisms by which exercise improves brain health and brain function. So what you can see here, if you look at the left-hand diagram, is we're looking at wild-type mice and Snell dwarf mice. Both males and females have more GPLD1. And the next shows for a different mutant, growth hormone receptor knockout, they have more of GPLD1 in their plasma. So that's a shared factor. But then we looked at the plasma of calorically restricted animals, that's CR, and at 4 different drugs: canagliflozin, acarbose, 17-alpha-estradiol, and rapamycin. WT stands for wild type. That's our control mouse. And you can see all 4 of those drugs and the dietary restriction also show elevation of GPLD1. The blue symbols are the males, the red symbols are the females. This is one of those that is not sex-specific. So we have 2 mutants, and in fact 5 mutants. I've only shown 2 here. 4 drugs and a diet, all of which elevate GPLD1. Oops. No, it's Chinese. Help. Let's see if I can get that. Okay, so I didn't show you the other mutants. It's true for the liver, it's true for the fat. This is only the plasma, so it's true wherever we look. Now, Villeda had shown that elements of brain health, BDNF and double cortin, also go up after exercise, so we looked at those in our mutant mice and in our drug-treated mice. The mutants are shown on the left, the drug and diet mice are on the right. Now we're looking not at the plasma, but at the brain. And BDNF, which shows helps protect brain cells from death, and double cortin, which is a marker of neuronal cell turnover, they go up also in all 5 of the mutants, all 4 of the drugs, and both of the diets that we looked at. So that is a shared phenomenon in all of the slow-aging mice. Then we turned to fat, and we looked at uncoupling protein 1. It's a protein in your mitochondria that helps you avoid fat accumulation and uses energy for heat production. And you can see in the brown fat, the inguinal white fat, and the perigonadal white fat, of the growth hormone receptor knockout mice, that's the top line, and the Snell dwarf, which is the bottom line. And for all 3 fat depots in canagliflozin, in calorie restriction, acarbose, and 17-alpha-estradiol, UCP1 is up. Rapamycin is the exception. This is the only exception in the talk today. We understand why that is, but I don't have time to tell you about it. But except for rapamycin, this change in fat biology is universal for the slow-aging mice. And it's not just UCP1. The white fat becomes brown fat. It becomes beige fat, and the cells have more mitochondria, less fat storage, all the sorts of things that are associated with excellent metabolic health. Now, the fat also is a source of inflammation, so we looked at 2 kinds of macrophages. M1 macrophages are inflammatory, and M2 macrophages are anti-inflammatory. If you look at the growth hormone receptor knockout mice, they have their anti-inflammatory macrophages going up, the pro-inflammatory macrophages going down. There's less inflammation in the fat. And that's true of the Snell dwarf, and it's true in all 3 fat depots, and it's true in all 4 of the drugs, and it's true in caloric restriction. So in the fat, 2 things are happening. There's a change in the macrophage subtypes, and there is an increase in UCP1 and a conversion of white fat cells into brown fat cells. This unifies all 10, and now also in the isoleucine story, all 11 of the slow-aging mice. There's a strong hint here that something upstairs, which we don't know, is causing the fat changes, the brain changes, and the plasma changes. Now, we do know that the fat change is probably due to a protein made in the muscle, and that protein is called FNDC5. A cleavage product called irisin goes from the muscle to the fat to do the good stuff. So we looked at FNDC5 using Western blots, and this shows the Snell dwarf and the growth hormone receptor knockout mice as we had imagined. FNDC5 is elevated in the tissues, the muscle tissues of those mice, and it's true for canagliflozin, calorie restriction, acarbose, 17-alpha-estradiol, and rapamycin. It's true for males and for females. So far I've shown you changes in the plasma, in the liver, in the muscle, in the fat, and in the fat-associated macrophages. This is the most technical slide that I'll show today, and if you don't understand it, that's okay, you'll get the point. But there's a special kind of protein translation called cap-independent translation. Most messenger RNAs are translated when the ribosome sticks to the 5' cap, and then it moves down and starts to make protein. But there's a small subset of mRNAs, special ones, that can be translated by cap-independent translation. And we had previously shown that it's elevated in all 5 of the mutant mice and by rapamycin. Rapamycin actually makes that change within 7 days after you give it to a mouse, and within 2 days in a tissue culture. Now, the really great thing, the most amazing discovery, is that the protein I was talking about, GPLD1, to our astonishment and pleasure, is a cap-independent translated protein. It is not changed because of mRNA levels, it's changed because of differential translation. So let's look at that. We're looking now at cap-independent targets. Acarbose, which extends lifespan significantly in both sexes, turns on many different cap-independent translated proteins in male and female mice. It doesn't change their messenger RNA, but the protein goes up anyway because the messenger RNA is translated by cap-independent translation. 17-alpha-estradiol produces a lifespan effect in males, but not in females for unknown reasons. And it turns on cap-independent translation in males, but not in females, again for unknown reasons, suggesting strongly that whatever mechanism upstream turns on cap-independent translation is an important element of the slow aging process. So there's a review in a new journal called Aging Biology. This chart is taken from that review. If you can't find it in your library, again, send me an email, I'll send you a PDF. But this sort of summarizes this year's version Obviously, I don't want you to read it or memorize it, but I do want to emphasize a couple of things. These aging rate indicators are detectable in liver, kidney, muscle, heart, brown and white adipose tissue, plasma, and hippocampus. They influence a wide range of single gene mutations that extend lifespan, and they also are seen in animals which are genetically normal but have been given an anti-aging drug or the calorie restriction diet. So the themes I want to emphasize is that these are not biomarkers of aging, and the reason I sound angry is that whenever my papers are rejected, it's because a reviewer says, biomarkers of aging, that's old-fashioned, they were doing that in the 60s. These are not biomarkers of aging. Biomarkers of aging are like the odometer in your car. They tell you how many miles your car has been driven. Biomarkers of aging tell you how many years your body has been driven in a sort of crude way. These are like the speedometer in your car. It tells you how fast your car is going currently, how fast you are aging currently in the aging sphere. If I tell you that your car is going fast, you don't know whether it's a new car or an old car. And if I tell you that your car has a lot of miles on the odometer, you have no idea how fast it's going. The aging rate indicator tells you how fast you're going. This is a cartoon showing what happens if you add a drug, some hypothetical biomarker which goes up with age, it'll now go up more slowly. Aging rate indicators are nothing like that. They change very soon after an anti-aging drug is added. They may go up, they may go down, it doesn't matter. The point is that they always discriminate at any age a slow aging mouse from a normal mouse. So the second theme I want to emphasize is that if you were someone who was working with RNA data, you'd miss this whole story. The CAP-independent translated proteins are changed by differential translation, not by differential mRNA production, not by differential transcription. And the third theme I want to emphasize is that many of these endpoints are not cell autonomous. It turns out that BDNF and double cortin in the head are modified by GPLD1 produced in the liver. GPLD1 in the liver is modified by changes somewhere else in a tissue we haven't identified yet. The point for those of you who are interested in drug discovery is you can't find these in tissue culture. Tissue culture, you're looking at changes in a single cell type. These things involve at least 2 or 3 different cell types talking to one another. These are missed by oversimplified models. So here's some questions and implications to leave you with. What we want to do in the next 5 years is to ask what mechanisms produce all of these cellular molecular changes in all 11 kinds of slow-aging mice. That's sort of the holy grail, the upstream thing that makes changes in all these tissues that reflect the slow-aging state. Now, downstream is pathology. That's the domain of molecular and cellular pathology. How do these changes produce delays in so very wide a range of age-associated diseases and cellular changes, including, of course, cancer and death and the things that worry us, but things that are less lethal but equally problematic. Logistically, I really want to know how long does it take a drug to switch these? Does it take, I know 8 months is correct. 8 months, the changes have been made. Can they be detected in 4 months? Can they be detected in 2 months? Can they be detected in 1 month? Once we know that, we can begin to screen new drugs. If somebody says, I've got 20 varieties of ergothioneine, and I want to know which of these variations might be most efficacious, instead of doing expensive lifespan studies of all these mushroom products, we can use these aging rate indicators in principle and pick the concentration, the specific isoform, the combination of drug that changes the aging rate indicators quickly. And we can also use this, even in people, to ask whether we can make these switches late in life. Now, we'd like to know whether this works in people. I kind of hope it does, but that's speculation. We don't have data. And we'd also like to know whether they're under genetic control. It's very likely that people who inherit genes to make them long live do so because they inherit genes that are making them have the favorable amounts of these aging rate indicators. And I will take my remaining 13 seconds to give you a pitch, an advertisement about the ITP. Any of you can suggest new drugs. One of the reasons the ITP works so well is that we don't have to come up with good ideas. We are dependent upon the scientific community to come up with good ideas they want us to test. If you want help with the process, look at the ITP website or send me an email and I'll help you. You can also take our drugs and test them in your system, whether it's a mouse-based system or a human-based system. If you want to know whether this drug modifies a mouse model of Parkinson's disease, that's great. You just buy the drugs from the same place that we buy them. And we have tissues from almost all of these mice. So if you have a molecular or cellular or biochemical question, we can send you for free the tissues from these mice as part of a research collaboration. When I told my humanities friends, my wife was an English professor, when I told her colleagues I worked on anti-aging medicines, they said I was a very bad person. They knew what old people were like, old people who were sick and grumpy and really unhappy, and they didn't like that. And they thought, incorrectly, that this was my job to increase the number of grumpy, unhappy old people. That is not the goal of aging research. The goal of aging research is to produce a new kind of old person. This is your typical average 70-year-old person today. It was kind of Harrison Ford to pose for this picture. And if these drugs did work in people, which we don't know yet, but if the drugs did work in people and work to the same extent that they do in mice, that would be your picture of your typical average 114-year-old person. I've tried to mention some of the collaborators who've done this work. I don't have time to mention them all and still stick within my allowed limits here. So this is our stress-resistant mouse model, the one that has been most successful and least applicable to pharmacological manipulation, but it's also a good slide to use for a question and answer period, so I'll leave it on and be glad to have questions.
感谢 Richard Miller 教授的分享，请观众朋友们踊跃提问。
Are there questions?
Hi, Richard. It's amazing findings from your ITP study.
Thank you.
I just wonder what separates the list of the drugs with benefits and the drugs without the benefits is based on the murine, like with heterogeneous genetic background, right? Okay. So to address the gap from mouse to human, I wonder, is there any human exposure database? Let's say if there are group of subject exposed to rapamycin versus the subject without, and retrospectively study their lifespan, any difference? Anything like that existing? I know rapamycin is probably not as popularly used as, metformin and so on. I just wonder, anything on your list with benefits has any clinical support?
I think the data has to come from a large-scale, placebo-controlled, double-blinded clinical trial of many years' duration. There are people, of course, who believe this stuff enough that they're willing to take chances. They have high risk tolerance. They're going to dump the stuff into their bodies and kind of hope that it works for them. And you can take materials from these volunteers and begin to ask questions, including now asking about aging rate indicators. That's the purpose of doing the aging rate indicator study. But there are some human databases that are pertinent and appropriately controlled. For instance, there are many people who are taking canagliflozin, not because they're diabetic, or empagliflozin, or the whole SGLT2 inhibitor family, but because they're pre-diabetic and they're hoping to postpone diabetes. And some of them can take it for a decade. So I would love to have plasma samples from individuals taking some of these drugs because a doctor thought it was good for them, and test aging rate indicators in those. It's difficult because, of course, most people taking these drugs took them because they have diabetes or because they had cancer or something, and that's a hopeless confounder. But you can imagine situations where chance data sets may exist that are informative, and of course, there are now excellent reasons to want to do 5-year studies of volunteers who are healthy. I think, and that's the point of the talk, of course, that the aging rate indicators can accelerate that process greatly if indeed they change within a year, say, of the time you go on a drug. You don't have to wait for aging to happen.
Yeah, great. Another quick question is that in one of the slides, you show different interventions achieve the same benefits, such as rapamycin calorie restriction, and maybe CNNA stands for canakinumab. So I wonder, does this suggest that all different pathways can achieve the same benefits? All these interventions share a common pathway?
That's exactly the correct interpretation, I think. That's the reason I wanted to give the talk. I believe that now that we have many drugs and diets and genes that all accomplish slowing of the aging process, for the first time ever, we now have the tools to ask that question. What is it upstream that is doing all of this? The work I showed says that, amazingly, they all change the fat in the same direction. They all change the macrophages, the liver, the brain, the muscle, etc., etc. And we're just scratching the surface. In 5 years, we'll have a much longer list of characteristics of the slow aging state. And then we can begin to ask, well, now, what causes that? What is responsible for shifting to the slow aging state? We have some unpublished mouse data, which I don't have time to share until the coffee break. I'll be glad to share it at the coffee break, that you can influence lifelong changes in these aging rate indicators by interventions for mice in the first 3 weeks of life. So there may be some things you can do to very, very young children that will have long-term permanent health benefits. But of course, the ITP drugs show that you can also change those. You don't have to do it in children. You can also do them by starting in middle age even.
Yeah, it's amazing, especially some of the interventions you're utilizing target specific pathways or particular targets.
Yes, that's the key point, that regardless of the target of rapamycin, the target of canagliflozin, the target of unknown target of 17-alpha estradiol, they're all different. So the key point is, what is uniting those? What is the thing that they are all influencing? There's a lot of good ideas. Some of the other speakers have mentioned good ideas, changes in redox potential, protection of lipids or proteins, or changes in DNA damage repair. There's lots of really good ideas, very little evidence, but lots of good ideas as to what those shared mechanisms might consist of at the molecular basis.
Thank you.
中间过道的话筒开一下。
Hi, nice, Richard. I would like to ask you about the statistical test, because we are, we are working on this kind of lifespan, and we do know that the log rank test is a common, but not the best at some point, especially if you see some kind of crossing the survival curve. And so just recently, that I think you're also aware of that. One of your collaborators, I think from, was that James Nielsen, they published to use another approach to analysis some of the statistical lifespan curve, and they found out there's some of things which before we mentioned that they're not going to extend the lifespan, but now actually found out they actually have some benefit, at least trying to squaring the lifespan curve at some point. So would you like to give some comment on that? And also in the future, if you like to also imply with another statistical approach, trying to find out or analyze some of the exist data for getting more information from the ITP program. Thank you.
So for people who don't know Jim Nelson's publication, which was also with Jonathan Gelfand, they were able to discover that some of the ITP drugs, these are using our published, publicly available data set, lowered your mortality risk when you're young, but increased your mortality risk when you're old. So they had a significant effect on mortality risk, but it was specific for different ages. And that's quite interesting. And there may be mechanisms that lead to death before the median lifespan in mice that these drugs influence, even though they don't influence the maximum lifespan or risk of mortality at later ages. So there is no one single number, a p-value that tells you what a drug did or whether it is good for you or something. It's a much more complex process than that. And in fact, we've noticed some drugs that have no effect on the median at all. When half the mice are dead, you can't tell whether drug or control, but then they start to kick in, then they start to work, and they produce an increase in maximum lifespan. So I'll just say, to provide information for when people want to test this, all of our data is publicly available, all 18 years of it. So if you have statistical tests that you would like to try on our data set, it's free, and you just ask for it, and you can have it. There's a pending publication from Robert Williams and his colleagues which will give nuance to this. He's found some segregating genes that influence lifespan, but only in the first half of life, and others that influence lifespan in our mice, 10,000 of our mice, but only in the second half of life, and others that are equally influential regardless of the age of the mice. There's more to the paper than that. Some are male-specific, some are female-specific. But in general, the baseline idea that if a drug works, it affects you regardless of what sex you are and how old you are, that's oversimplified.